Hello children, yesterday we have learned about the Mendel and the why did he selected the pea plant and his experimental procedure. See, even though he did his experiment, it's such a nice way, he conducted the experiment and gave us some of the principles which we will discuss little later. But then also he failed. During his lifetime, he did not come to know the recognition of his work. So today we will see the reasons for Mendel's failure and after his death, why he got the success, why he got recognized by all of us. So let us see the points. So reasons for the failure or unrecognition of Mendel's work. See, Mendel published his work in 1865. 1865, it remained unrecognized till 1900. So what were the reasons for his failure? So the first was due to the low level of communication. During that time, the communication was not much. So he published his work in local magazines. To get the recognition, one must publish their work in international level. At the international level, during that time, he published locally, and limited number of people were knowing about his work. So this is also one reason. And then his concept of genes. During his time, he called genes as factors which are stable and discrete units and were responsible for the transmission of characters from the parent to the progenies. And this was not accepted by many scientists because he did not provide any proof for the existence of these factors, and many were showing the intermediate characters. So he did not explain those one. That is why many scientists did not understood his concept of gene. And then he used mathematics in maintaining the experimental data. He used mathematics in maintaining his experimental data. So during that time, use of mathematics in biology was not much. So this mathematician explaining biological experiment was not accepted by many of the scientists. So these are the three important reasons why Mendel failed. Mendel's work did not got recognized during his time. So that is about the reasons for the failures. What were the reasons? Publication problem. That is, he published his experiments in a local magazine and limited people were knowing, and that too they did not accept it. International level of publicity was not done during his time. And then the concept of factor, which are now we will say them as a genes. They are stable and discrete units and they carry parental characters. The transmission of parental characters to the progenies are done by these genes. So he did not provide any proof for their existence. And then he used mathematics to maintain his experimental data. So these are the three important reasons for his failure. Now after his death, three scientists independently worked and rediscovered Mendel's work. So they were the Hugo de Varies of Holland, Schirmack of Austria and Karl Correns of Germany. These three scientists rediscovered Mendel's work and made him to get the credit of his work. So the thanks must go to these three scientists who made John Gregor Mendel as the father of genetics. And now also we will follow the genetics basic principles which Mendel gave us. Now let us know why Mendel got success. So reasons for success, or you can say recognition of his work. The first one is the selection of pea plant. So he selected pea plant for many reasons which yesterday in previous class we saw why Mendel selected pea plant. And now in that also he selected limited contrasting characters, which is very important. He selected 7 contrasting character. There was no intermediate character between the characters which he selected. Like tall means it is tall for one, dwarf means short for all. There is no in between character. Likewise, the purple or violet color. Yesterday I wrote violet as V, even we can write purple, capital P and small p. Yesterday I used V for violet. So in book it is given purple, so let us consider that as a purple. Okay, there is no intermediate character which he selected, and that is a plus point. That is a credit we can say. I mean, the selection of limited contrasting characters. He selected only 7 characters which were contrasting. So that he did, and use of mathematics and statistical data in maintaining his experimental analysis. He analyzed all the experimental data very carefully and represented them in a mathematical way. He analyzed them by using the knowledge of mathematics and statistics. So that is also a plus point. And also it is a very, very important point that the 7 characters which he selected did not show any linkage. Otherwise, there would have become a problem for proposing the principles of genetics. So these are the 4 reasons for his success. What are the 4 reasons for the success of Mendel's work? The first one is the selection of pea plant. Why selection of pea plant? He did that we saw yesterday. And then selection of limited contrasting characters. At a time he did not took more characters. At a time he did not consider more character. 7 contrasting characters he considered. And then he used math and he followed. This is also very important. While doing the experiment, he followed the same stepwise procedure. That is 3 important steps he followed. Selection of pure line, hybridization, then selfing. So those 3 experimental steps he followed. And then he used mathematics in maintaining biological experimental data. And then the selected characters by him did not show any linkage. Because of all these reasons, he got success in proposing the principles of genetics. Now let us see some of the important terms which oftenly we use in genetics. Now let us know the terms. Now let us see the terms which we use oftenly in the genetics. So few terms I wrote here. Chromosomes. The first one is the chromosomes. They are the vehicles of heredity. They are the vehicles of heredity which are transmitted from parent to the progeny generation after generation. So that is about the chromosome, and the structure of chromosome you have studied in first year, and in second year also you are going to study in coming periods in detail. Now next is the genes. So genes are the functional units of heredity. They are the functional unit of heredity responsible for the expression of a character. They are the functional unit of heredity which are responsible for the expression of a particular character. Now next is the alleles. So alleles are the pair of genes present on the same locus on a homologous chromosomes which will control the alternate characters of a same character. That is alternate expression of the same character. That is about the alleles. Or simply you can say they are the alternate forms of a gene. They are the alternate forms of a gene which are present on the same loci, at the same point on the homologous chromosome, and they control the alternate expression of a same character. Same characters, alternate expression, it will control, or simply you can say alternate forms of genes. And locus. Locus, it is a specific point or a region on the chromosome where genes or alleles are present. That is called locus. Now next is the hybrid. Hybrid, it is an individual. It is an individual which is obtained by crossing two different individuals which are different in genetically they are different. So such an offspring is called hybrid. What do you mean by hybrid? An individual obtained by crossing two genetically dissimilar organisms. That is about the hybrid. Now phenotype. Phenotype, it is the external expression of an organism. That is external appearance. The external appearance of organism is called phenotype. Like in the height we will express tall and dwarf. Only one example I have taken. Genotype, it is the genetic constituent of an organism, or you can say the genetic component of an organism. Like in the case of tall, capital TT, and in the case of dwarf, small tt. So genotype, I did not write here the genotype. Now let me write. So capital TT, it refers to tall, and small tt for the short. So that is about the genotype. And pure line organisms or breeds are those progenies which are obtained by crossing the genetically similar, or you can say genetically pure homozygous true breeding individuals, and we obtain these through continuous self-hybridization, selfing. So pure line organisms are those organisms which are obtained by crossing two pure homozygous individuals. Through continuous selfing we will get, that is the pure line organism. So next, hybridization. So hybridization, it is a technique which involves in the formation of a hybrid by crossing genetically different organisms. What do you mean by hybridization? It is a technique by which we will get the hybrid. How do you get the hybrid? By crossing two individuals which differ genetically. That is about the hybridization. These terms you have to learn, because oftenly we will be using in the genetics. Like what are they? Let me just repeat. Chromosomes are the vehicles of heredity. They are called vehicles of heredity because they will go on passing from generation to generation, and they are responsible for the expression of the character. Genes, they are the functional unit of heredity. Alleles, they are the alternate forms of the genes which will control the alternate expression of a same character. And next, locus, it is a specific point on the chromosome where genes or alleles are present. Hybrid, it is an individual which is obtained by crossing between two individuals which genetically differ. And then phenotype, it is the external appearance of an organism. Genotype, it is the genetic constituent of an organism. Pure line organisms are those organisms which are obtained by crossing two pure homozygous individuals, that is called pure line. We will get it through continuous selfing. Hybridization, it is a technique of obtaining hybrid. How do you obtain hybrid? By crossing two individuals which are different genetically. Next terms, let us just see the next terms. Now let us know the next term, that is the homozygous. Homozygous means presence of similar pair of genes on a homologous chromosome. That is, the genes which are present on the homologous chromosomes are same. If they are dominant, then both the homologous chromosomes must contain the dominant gene. If they are recessive, they must contain the recessive genes. Like capital T, capital R. Here I gave two examples. So that is about the homozygous condition. What do you mean by homozygous condition? Presence of similar genes on a homologous pair of chromosomes. Now heterozygous condition. The name itself indicates different. That is, heterozygous condition, it refers to the presence of two dissimilar genes on a homologous chromosomes. So that is called the heterozygous condition. Here I give the example of capital T, small t. That is the one gene which controls the tall character and the other gene which controls the dwarf character. But here of course the dominant character is expressed. But as both the genes are present on a homologous chromosome, this condition is called as heterozygous condition. Now dominant gene. In previous class we saw what do you mean by dominant. A dominant gene is the one which expresses its character under heterozygous condition. So that is called the dominant gene. So here we have taken the tall and the dwarf as an example. Here the homologous chromosomes contain both the genes, that is tall and the dwarf. But it expresses only the tall character. The gene which will control the tall character is called as dominant gene. Now recessive gene. Recessive gene is the one which is not expressed under heterozygous condition. That is, it will get suppressed. Its expression will get hidden in the presence of the dominant gene. Such a gene is called as recessive gene. So here we have taken the example of, let me take this only, capital T, small t. So here tall character is expressed. Dwarf character did not got expressed. It got hidden. It got suppressed by the presence of the dominant gene. So the small t, the dwarf character which is controlled by small t is called recessive gene. So here the small t is the recessive gene. And now next is the test cross. Test cross, it is a method to know the genotype of a hybrid. So here the cross is made between the F1 hybrid and its recessive parent. It's recessive, homozygous. The recessive of course is a homozygous condition. The recessive characters are expressed under homozygous condition. The cross between the F1 hybrid with its recessive parent is called test cross. Why this test cross is done? So by this we will be knowing the genotype of the hybrid, whether it is homozygous or heterozygous. Now next is the back cross. So it is a cross between the F1 hybrid and one of its parent. So one of its parent, just here to increase the characters of their parent. So that is called the back cross. What is back cross? F1 hybrid is crossed one of its parent. Why this cross is done? Especially to increase the number of characters of the parent in the progenies. So that is about the back cross. Now monohybrid cross. Cross is hybridization. That is breeding of two individuals to get the progenies. So monohybrid cross, it is a cross between two individuals of the same species considering the inheritance of one character. Considering the inheritance of a single character is called monohybrid cross. So in hybridization experiment, when we consider only one character, when we concentrate on one character, that is called the monohybrid cross. Dihybrid cross. So it is a cross between two individuals of the same species. Between two individuals of the same species, when we consider two characters, when we consider the inheritance of two characters, then that cross is called as dihybrid cross. So what all terminologies now we have learned? Homozygous. What is homozygous condition? It is a condition where paired genes on the homologous chromosomes are same. So these are the questions which are asked in annual exam. What example do you give? Capital T, capital T. Let us take this as a common example. Height as a character for giving the examples for these terms. So homozygous I told. What do you mean by homozygous? Presence of similar genes on a homologous chromosome. And heterozygous condition, presence of dissimilar genes on a homologous chromosomes. And the dominant gene, it is the gene which is expressed under heterozygous condition, and that is always indicated by the capital letter, and example we gave here the capital T and small t. Here this will express the tall character which is controlled by tall capital T. So this capital T is the dominant gene. Now recessive gene is the gene which is not expressed under heterozygous condition. So it is hidden under heterozygous condition, or you can say in the presence of dominant character it will not get expressed. Such a gene is called as recessive gene. The above case only we have taken as an example. Here dwarf character got suppressed by the tall character. So small t is a recessive gene. And then test cross, it is a method where F1 hybrid is crossed with its recessive parent to know the genotype of F1 hybrid. So that is called the test cross. Back cross, it is the cross between F1 hybrid and their parent to get more and more parental characters. Monohybrid cross, it is a cross between two individuals of the same species by considering inheritance of single trait, by considering inheritance of single character. And dihybrid cross, it is a cross between two individuals of same species considering the inheritance of two characters. Mendel called characters as traits. So in the book usually we will see the character referring the term trait. So let us see some more terms. Now let us see the next term, emasculation. This term you came across in reproduction in flowering plants, and even yesterday when I was saying you the procedures or steps involved in Mendel's experiment. We have seen yesterday the experimental stages of Mendel which he followed during his experimentation. There also you came across this term, emasculation. What do you mean by emasculation? It is the removal of male reproductive structures from a flower before maturation of anthers. Simply removal of anthers before its maturation from a bisexual flowers. That is the emasculation. Incomplete dominance. So here the dominant gene is not completely dominant over the recessive gene, or either of the two alleles are completely dominant or completely recessive. Incomplete dominance I was saying. That is the dominant gene is incompletely dominant